And congratulations on your pronunciation of Aberystwyth. Most people can't do that.
I worked on it.
So I'd like to thank the opportunity to present again at this meeting. And again, similar to Chris, I'll be expanding a bit on the last presentation and showing you some of the new results we have. And as I realized when I was looking at the program, I'm sandwiched between two bone talks. I thought I would stick to mainly towards cartilage, which is between the two bones. And the exciting sheep project is the one that comes after me, so I have no sheep at all. So for those not familiar, this is our research institute, and this is where we live and work. So, and the AO Foundation is one of those, just quickly, the AO Foundation is one of those foundations which nobody's ever really heard of, but everybody knows what we did. And so what the AO Foundation is actually famous for is the development of all these plates and screws and nails and pins which are used to fixate fractures when you have a traumatic injury. And so based on that, we've been working along how trauma can affect patients and how can we basically bring patients through to a quicker healing and a more effective healing. And so one of the main topics of this particular presentation is going to be articular cartilage. It's the surface between the two bones. Everybody's very familiar with it. It's got a single cell type, although now there's a lot more evidence that there's actually a progenitor cell population within the cartilage. And it's one of the more difficult tissues in the body to heal. It's very, very hard to heal cartilage. So often if you have an intra-articular fracture, for example, the bone will heal fairly well, and it's actually the cartilage which gives the problems later on. And from a tissue engineering perspective, this was always considered to be an easy target. There's one cell type, there's no blood vessels, there's no nerves, there's no lymphatics, so it's quite a simple tissue. And yet it's proven very difficult. And so when you look at some of these repair strategies, how do you fix a traumatic injury within an articular surface? So you imagine you've fallen off your bike or something, you have a hole in your cartilage. One of the more clinically applicable treatments, which is currently applied in patients, is this idea of this autologous chondrocyte implantation. You take the cells from the patient's own cartilage, you grow them up in culture, and you reimplant them under some kind of flap or a tissue flap. One of the more simple methods and one of the most commonly used methods is this microfracture method. And essentially what happens there is you have the defects, it's debrided, you cut away the edges to make it clean, you puncture the bone, and bone marrow moves into the defects, and it brings with it marrow stromal cells, so bone marrow MSCs. Now, one thing to bear in mind when you have this microfracture is you have human cells in a fibrin clot, which are then the bone marrow-derived stromal cells. And the main problems with these are that it doesn't really produce usable cartilage. So this is believed to be more robust than this. And so bone marrow stimulation, although it's commonly used, is actually quite a poor treatment. And so although everybody started thinking of tissue engineering, this is a very simple tissue and it's going to be very, very easy to repair, what we're beginning to realize over the years is it's actually quite tricky to repair. And some of the questions of that are why. And when you look, for example, with most of these treatments, the previous two, for example, one is using chondrocytes and one is using MSCs, and yet there's no standardized rehabilitation protocols, and often both sets of patients are treated in a fairly similar way, because it's just a cartilage regeneration. And so coming back to this idea, when you're using this stimulation technique, the mesenchymal stem cells or stromal cells we know can undergo chondrogenesis. And so there's been a lot of work over the years of how can you turn those bone marrow cells into chondrogenic cells. And the most standard in vitro protocols we use is a very simple cell culture technique. You would take static culture, you would put your MSCs into a hydrogel of some kind, a three-dimensional environment. You would add TGF Beta, transforming growth factor beta protein, and that's giving you this red stained, the same safranine from the previous talk, cartilage. And the one thing about that is it's very simple, it's very easy, but it completely decouples mechanics from biology. And we know with these kind of tissues, mechanics is crucial. So how much of this general assumption is true? How robust is that model? The second thing, when you started to look at these models using bioreactor systems to apply load, is we know from many studies that if you apply uniaxial compression, so you basically move a piston up and down, if you embed chondrocytes into a gel, They build their new pericellular matrix because they're chondrocytes. Then when you start to compress them, they start to respond like chondrocytes and make more matrix. So a lot of studies which started to look with the MSCs for mechanoregulation used exactly the same bioreactor systems, which kind of suggests that the signal for chondrogenic maintenance, the physical signal, is the same for chondrogenic induction. So that's another question. Load is very complicated. So is that actually true? Now, so we've been looking for a number of years at these biological systems, and we're very interested in how the mechanics drives the biological response. And so for that, we think you need a physiological load. So you need to be able to apply a load which you can apply within a patient. Because if you want to use this as a rehabilitation protocol, you need to be able to do something patients can do. So you need a scaffold material if you're going to think about scaffolds to enhance the repair. You need a scaffold material which can accurately transmit the load you're applying. Some of the ceramics we use in bone, for example, they're rigid. They don't apply, you can't apply load to them very easily. You need a biologically relevant source of cells. So we only use human. All the presentation today will be just purely human cells. And the reason we believe that's true is because when you're using young animal cells, they're epigenetically programmed to grow. So they will make more matrix because they're very, very young. Whereas the adult cells you're trying to treat, they're epigenetically printed not to grow, so they're maintenance. So that's sometimes why we think that the animal studies don't translate very well into humans. And you also, again, as I mentioned already, you need to have a protocol you can actually apply. So we've been working on this machine now for a great number of years, and our bioreactor is pretty complex. We have a ceramic hip ball which is implantable into patients. And so we can move the hip ball up and down for compression, we can rotate it for shear, or we can move the holder to completely mimic the type of articular motion you might see in a patient's environment. And most of the work I'll talk about today is we've used this standard protocol of plus minus 25 degrees, 1 hertz, and then basically 10%. So 1 hertz is an injured patient trying to walk around a bit slower than they normally would, for example. And when you do this, and when we started many years ago with this PhD student, Sen Lee, normally coming back to that static culture model, people add 10 nanograms TGF beta to induce chondrogenesis. And when we do that, looking at these two markers, aggrecan and collagen 2, very classical cartilage markers, we didn't see any response to load at all. And so at the time we were doing this, that didn't make any sense whatsoever, because everybody will tell you musculoskeletal system is heavily regulated by mechanics, and yet we consistently got data which told you that it's not regulated by mechanics at all. So then we started to think, well, okay, well, where does the TGF beta come from within a patient? And it turns out if you take the TGF beta out of the system, you now have a very robust response to load, and you can start to induce chondrogenesis in the absence of proteins. So this culture medium now has no serum, it has no growth factors. We have elderly or adult human bone marrow cells in a fibrin gel, which was clinically available, and then we started to induce chondrogenesis purely by mechanics. And so for all the studies I shall be showing from now on, has no exogenous proteins, no TGF. Now, just touching on bone, because I am from the AO, I should mention something about it. And it was what Chris mentioned in the previous one. We know that there's this mechanical regulation of MSCs. We know if you rigidly fix bone, it goes straight towards an osteoblast. It does not have a cartilaginous template. Whereas if you have a mechanical environment, you push it towards a cartilaginous template, you get this micromotion, which is what the aspect of this reverse dynamization. And so if you can control that mechanical environment, you can control this indirect versus direct healing, exactly what happened in the last presentation. And when you look at models of how this works, what you'll see is that over time, this material, which is in a fracture, is the same kind of hematoma material because it came from the bone marrow, which would be in the fracture, in the microfracture cartilage treatment. And what this model says is if you have too much motion, you get a fibrous non-union. If you have micromotion, you get cartilage. And if you have no motion at all, you get bone. And so this then starts to question, again, the kind of static culture models we're using for cartilage, because we're pushing these cells, we're pushing towards cartilage with a growth factor, but then we have them in a rigid environment. And one of the biggest problems we have when we do this in culture is that hypertrophy. So the cartilage we're forming is not stable. And maybe, again, it's due to this mechanics. But what this model also would predict is if we change the frequency and amplitude of the load we applied to the cells, we should be able to push the cells more towards cartilage and more away from hypertrophy and bone. And when we did that, you see exactly what the model predicted, that as you increase frequency or amplitude, you increase aggrecan and collagen II production, so you're pushing it more towards a chondrogenic phenotype. But the question then becomes, is it just increasing all gene regulation because you're increasing fluid flow, or is it actually shifting the phenotype? And one of the markers which in resting cartilage you do not want to see is this collagen 10, which is a classical marker that these cells will go on to form bone. And when you start looking at the ratios of the genes you want, like aggrecan, and dividing them by the genes you don't want, what you see is exactly the same pattern, that as the frequency and amplitude increases, you're pushing the cells more towards resting cartilage and more away from hypertrophic cartilage. And that was the same for the different collagen ratios, so we were quite happy that the model we have seems to be working in the real world as well as just in vitro culture systems. So where we got very interested then is start to dissect the mechanism of action. How does this actually work? And for rehabilitation protocols, this becomes relevant because you can apply just compression. You can apply a brace onto a patient so that they can't move, they can't bend, but they can weight bear. You can give them CPM so they can have just shear, or you can have both at the same time. And so we turn the various motors on and off, and when we do that, what we found was that basically you needed both. So compression alone did absolutely nothing whatsoever, which was a bit surprising to us, and we're still trying to work out exactly why. Maybe I should qualify that in this system with this scaffold and these cells. Shear has a tendency to do something, whereas compression and shear is what we were seeing for the big increases in these ratios in the individual genes and this push towards collagen 2 over collagen 10 or ALP. So essentially, in our model, you needed both types of load. And when you look at the histology of that, it becomes fairly comparable to what you would expect from the gene expression. Essentially, what we're looking for is this kind of metachromatic staining of cartilage matrix, which only appears when you have compression and shear. And so I'll come back to the mechanism in a bit, but when you start now thinking about load versus static cultures, we collaborated with a group in Leuven, where essentially now we modeled our system. And what you can find with these mathematical models is different strain patterns. And now it becomes extremely clear that depending on which cell you are and where you are in this matrix, it makes a big difference to the type of load you sense. And so what we were doing was the modelers were coming up with very large numbers of these kind of strain fields. You can have all sorts of different numbers and things. But the question is, which is the one which makes the difference? And when we were looking at this, what we did is they produced a load of strain fields. We got a technician to sit down with a load of past historical histology sections, and she also graded them like a heat map to matrix staining for glycosaminoglycans. And then what we tried to do is correlate the histology to the models being produced by the FE simulation. And what we found was that this principal strain field actually matched quite well. And the nice thing was that compression alone didn't really do very much. Shear alone had some peaks where you could start to see increases in strain, but compression and shear robustly was transmitting these kind of loads into the scaffold material. And then the other nice thing for us was this model was produced by comparing glycosaminoglycan staining to the model. When we looked at the PCR data, coming back to this collagen 2, which is the collagenous model, it actually matched the collagen model very well as well. So we think we're trying to develop this model now because we believe we can use it for materials design. So you use the FE model, design your material in the model, and then try to reverse engineer the optimal mechanical properties, with the idea that when you do microfracture, you could do a patient-specific implant, put it into the defect, and transmit the loads better to improve microfracture or nanofracture. And again, now we know that where the cells are makes a difference, then it becomes quite clear that if you redistribute where the cells are, you should also be able to make a difference. And so with the PhD student, Oliver Gardner, we did the classical tissue engineering approach of evenly distribute the cells. We did a second group, which is 10% of the cells are on top and 90% are in the middle, but there's no difference in number between group 1 and 2. And group 3 was just a control of 10% on the top. And when you do that, when you induce mechanically loaded chondrogenesis, essentially what we got in every patient was exactly the same what we'd expected to see, that we got a much, much more robust histological outcome with a much more robust cartilage formation just by relocating the cells. And so when you want to know how this works, we started looking, of course, at the TGF beta protein, because that's one of the things we add. And when you do that, when we load, we find that we produce about 1 nanogram TGF beta from the cells, and that's about the same as when you add exogenously 1 nanogram. But when cells make it, they produce a latent form of the protein, whereas when you add it into a culture medium, you add the active form. So a question we had then was, can we activate it? How is the protein activated? And when you do that with our loading protocol, we can activate the protein itself. And so it's not just a production issue, it's an activation issue. And what we also realized was that when you activate the protein, you can do it in the absence of cells. So this is where we set up exactly the same system. The scaffold material, no cells, we added latent TGF beta, and then when you mechanically stimulate it, you can activate the protein physically. And so we're beginning to see that this is a way to recouple the mechanics to a biological response because it's a mechanical activation of a protein. And we know this is stiffness related. And so now we've been testing new biomaterials to put this into patients. One of our standard first protocols is we will add latent TGF beta into a cell-free system and see when we load, can we turn the protein on? And if we cannot do that, we know that that material, we think, will not be so good when you implant it into a patient. And then you can do the same with the cells, and you can see the increase in active protein. So again, looking at the biological response, more and more people are looking at secretome type models. How does the secretome, what the cells produce, change the behavior? And so one question is then, is this multiaxial mechanical load and TGF beta stimulation, is it the same thing? Or can we look for new molecules by applying load and see what differences we see? And so when you compare the load to TGF beta, looking at the protein level, you see multiple different proteins which are upregulated, and some with TGF, some with load. Then if you look at them, the ones in green are upregulated under both conditions, the one in red are differentially regulated. So you can see that there are different proteins upregulated depending on what the type of induction you do. And again, when you compare load alone, we started to find, and this array only had 174 proteins on it, and we already found a number of candidates which could be clinical targets for mechanically stimulated cells. And the PhD student doing this work suggested that when he looked at the protocols that the nitric oxide might be involved. And so when he looked at when he went back, this is one of our new targets at the moment, because every time we loaded the cells, we had nitric oxide being produced, and this nitric oxide is normally considered a very bad thing in the joint. So we might be able to, we're trying to work out at the moment, can we trigger the TGF beta protein activation without triggering nitric oxide synthesis? So he was very happy that worked. So just for the final couple of slides, coming back to this bone marrow stimulation, we have this the stem cells which everyone's been focused on, but as we've been hearing more and more during this meeting, we have macrophages in there as well. And strangely enough, there's very little known about the mechanoregulation of macrophages. And so you've seen this kind of work where this classical M1, which is needed at the beginning of a healing process, and then it transitions more into this more reparative, regenerative M2 phenotype. And so how is that controlled, or is it even controlled by mechanics at all? And the reason why this is important is because this is like a fracture healing model, it's a general healing response. This initial M1 inflammation is actually needed. It's like a danger signal to tell the body there has been an injury, but it does need to resolve and it needs to become an M2. And M2 can become M1, but M1 is believed cannot become M2. So the timing becomes very important. And so what we did was we took a number of we took human monocytes, so the precursors of macrophages, we embedded them in a gel, and some were differentiated or pre-differentiated for 3 days to see could we manipulate from one direction to another direction, and then we loaded for another 3 days and quantified to see what the response was. And so there's a lot of data on this slide, but you don't focus on the individual factors, just focus on the trends. And what you see is this M0, which is non-stimulated, so it's naive monocyte. If you load them with our protocol, basically what happens is you start to see at the mRNA and the protein level quite a lot of upregulation of pro-inflammatory markers. If you take the M1, which is already stimulated to be pro-inflammatory, basically there's very little difference because they're already quite active. But the interesting thing for us was that when you take the M2, which is supposed to be pro-healing, and you mechanically stimulate them, we also start to get, even at the protein level, a re-upregulation of these pro-inflammatory markers. So we're still trying to work out whether there's a sweet spot. This was quite an aggressive protocol because we didn't know if it was even going to work, so we didn't want to miss anything. And so now we're trying to see which kind of protocols you could use to optimize the healing response or change the healing response. But this could also play a role, as Chris just mentioned in the previous talk, about the mechanics, the regulation, and the immune regulation of the healing response. So with that, I've gone over a little bit, but so basically what we're trying to show is that we can couple the mechanics, so basically patient rehabilitation protocols, where, for example, there is TGF Beta in synovial fluid. You can activate it by the mechanics, and the induction and maintenance require different stimuli. And using this kind of rehabilitation work, we started to identify new markers. We started to see that we can modulate the inflammation, and we can try to dissect exactly how that's done. And we're now starting to look more and more into secretome. And so with that, I'd like to thank a lot of the PhD students who did the work and our clinical collaborators for the samples. And if there's time, I'll take some questions. Thank you very much.
Thank you, Dr. Stoddart.